Hi friends, welcome back to my channel Only Bio, presenting up the revision of the chapter Biotechnology which has been started. Hope you have been following this series. So friends, in this biotechnology which becomes the sixth part of the chapter, we are going to discuss something about r-RNA. So let's understand ribosomal RNA. On the screen, you can see a picture depicting up the different types of RNA. Friends, just to remind you of, there are three main types of RNA, that is mRNA, rRNA and tRNA. And just to remind you, these all are the subtypes of the non-genetic RNA, right? And they all are produced from the DNA itself in the nucleus. What I mean to say is that they all are produced in the nucleus from the DNA, from the two DNA strands. Only one that contains the information for a particular protein will be selected and would act as a template for the formation of RNA. So RNA polymerase enzyme will play a role. Now there are a family of RNA polymerase enzyme, and depending on which enzyme plays a role, what we get is either mRNA or rRNA or tRNA. Now they have been classified as mRNA, rRNA, tRNA depending upon the functions they perform. Friends, just to remind you of, these are the non-genetic RNA molecules, right? So it constitutes about 80% of the total RNA content of the cell. The base pairs of r-RNA are complementary to that part of nuclear DNA from where they are produced. Again, you have to remember, instead of thymine, what we would get is uracil, because this is RNA. r-RNA consists of a single strand folded upon itself at some regions. The folded regions have paired bases. What I mean to say is that the region where the r-RNA folds, so if there is adenine on one side and opposite there is uracil, so they will form a bond. And this area looks like double stranded only in the paired region or in the folded region, whereas the other regions will show the unpaired bases. r-RNA does not show equality in purine and pyrimidine bases. r-RNA are huge molecules, that is macromolecules with several hairpin-like intramolecular loops. Friends, this is not to be confused with the hairpin model of tRNA, okay? The r-RNA is responsible for the structure of ribosomes, in which they are complexed with many proteins, because it's a ribosome where protein synthesis occurs. They play an important role in translation of message contained in the mRNA. Friends, hope you like this. Please subscribe if you haven't yet done it, and hit the bell icon if you haven't done it. Thank you so much for being with me.